morning and good afternoon to all of you for joining our live webinar today, Structure and Function of Sodium and Proton-Dependent Transporters. Today's webinar marks the fifth and final talk that has made up our 2021 Research Insights Transporter webinar series. My name is Jason Villagomez, Marketing Manager at NhaA Technologies, and will be your moderator for today's event. But before we get started, my colleague Dr. Maria Barthelmess, Senior Scientist and Product Manager of the SURFE team, would like to say a few words.
Thank you, Jason, and welcome everybody also from my side and on behalf of my team colleagues, Andrea and Cecilia, who some of you might know. Thanks for joining us today for the final session of our Transporter webinar series. And a very special welcome to David and Matthias, our speakers today. It's really a pleasure having you, and thank you for sharing some of your work with us today. If you missed one of the previous sessions of this webinar series, you still have the chance to watch it on demand. You can find the links on our webpage under webinars. Because this is our last session, I want to take the opportunity and say a very special thank you to everyone involved in making this webinar series possible. So in particular to all the speakers, we really appreciate that so many people were willing to share their research here with us. But also thank you for participating. So there were really a lot of people joining us for these events. And you brought this event to life with questions and discussions. We hope that will be the same today. It takes some effort to prepare these webinars, and it's really a pleasure that so many people join. We would appreciate it a lot if you could give us some feedback or input about this webinar series. So please let us know if you enjoyed this format or if you would be interested in us continuing this webinar series in 2022. We would also be interested to hear about specific topics or sessions that you would be particularly interested in, or maybe you would even be interested to join us as a speaker. So we would be really happy to hear about that. You can write anything that comes to your mind right into the meeting chat today. It's not public, so just we will see that and we just use it for improving our webinars. And we will also send around a survey link after the session. So we would be really happy if some of you could take that and give us some feedback so that we can improve what we are doing. Finally, if you got interested in the technology and want to try it out yourself, or if you are already a user of SSM-based electrophysiology, then you can contact us anytime and discuss with us. And yeah. So with that, I hand back to Jason, and I'm looking forward to the talks.
Thank you for Maria for the recap. It really has been a wonderful year of presentations so far, and I'm looking forward to what's to come next. Joining us today as presenters are David Drew, Professor in Biochemistry from Stockholm University, and Matthias Quick, Associate Professor of Neurobiology, Department of Psychiatry and Physiology and Cellular Biophysics, Columbia University, Irving Medical Center. So a little bit of due diligence. Upon completion of the presentation, we'll hold a live Q&A session with the presenters, and as such, welcome you to submit questions at any time throughout the course of the presentation. You can log a question in the chat window found on the right-hand side of your screen. Your message will come to me directly and will be added to the queue, which we'll then address at the conclusion of the presentations. If you happen to experience any technical difficulties at any point in time, please inform us via the chat box as well. Additionally, today's presentation will be made available on demand shortly upon completion of the event, inclusive of a transcribed copy of the Q&A. I'll now turn over the presentation to Matthias for the start.
Okay, thank you, Jason, Maria. Can I share? Okay, should be able to see my screen. So thank you for inviting me, having me at this, I think it's the third event that I'm participating in the NhaA family of events. So I'm happy to be here today. I'm talking about the molecular mechanism of the sodium proton antiporter NhaA, from binding to flux. As you can see here, this is a view out of my window in the evening. Right now I'm sitting in my office, and what you see behind me is the morning sun, and I hope there's not too much glare. So if you have any issues, I try to move away and just let me know if something comes up. Anyways, I was preparing this talk, and I determined this to be the molecular mechanism, but I realized it's actually better if I change this title toward the molecular mechanism, because I don't think we have the molecular mechanism, so there's still work to do. And this is part of the seminar to basically use technology to come closer to getting a better picture of how NhaA and other proteins work. So basically, my background is in membrane transport biology. I've been working with basically all types of proteins that do either passive transport, active transport, or grouped translocation from channels, primary transporters. But my favorite, or what I call my home, is the secondary active transport field, where the symporters are located basically in its family. I started as an undergrad student in Germany at the University of Osnabrück, where I started working with a protein called PADP, which is the sodium proline transporter. This protein was cloned in the 70s, 80s and was shown to be a proton-dependent protein transporter in the beginning. However, it turned out that the proton dependence was not a direct mechanism. It was actually the action of a sodium proton antiporter, in which case, when you drop the pH of your medium, the sodium proton antiporter, in this case it's NhaB, starts pumping in the protons in the cell and exchanging this with sodium. Sodium is cycled back, and PADP can use the sodium ions to drive the protein transport. The amazing thing about PADP is the affinity for sodium is in the 30-40 micromolar range. So any kind of contamination or any sodium that comes into the medium unexpectedly will ruin your experiment. And in this context, I learned to work with sodium-dependent transporters. Be very careful to avoid any contaminations of sodium in my assays. So just to give you a short summary of NhaA. So NhaA it's a sodium proton antiporter of E. coli. It's the paradigm of sodium proton antiporters. This is a family that's conserved in all kingdoms of life. Their function is the homeostasis of the cellular pH, sodium concentration, and volume. Some of them in the mammalian family groups are implicated in diseases. But the NhaA was cloned in 1988 in the group of Etanapadan. It's a member of the cation proton antiporter CPA superfamily, specifically in CPA1 subfamily. It mediates the eclogenic exchange of protons and sodium, and you can also replace sodium with lithium. So you can also see proton lithium antiport. The flux stoichiometry is 2 protons by 1 sodium, and NhaA has a strict pH-dependent transport phenotype. And what's important here, this protein is active at a high pH. So at alkaline pH, NhaA has its peak of activity. It's basically inactive at pH values that are below 6.5, like 7 to 6 in this range. You see almost no transport activity. NhaA is amazing in the fact that it has a very high catalytic turnover number, which is in the 1,500 per second, which is for most of the secondary transporters really high. We see mostly cases from about 1 per second, some cases 50, but more than 1,000 per second is really pushing it close to the channel activity level. Not that it functions as a channel, I'm just saying the turnover number is going into the channel activity range. The structure of NhaA was determined with X-ray crystallography and also by cryo-EM. One of the features, as I mentioned before, is shown here. So this is NhaA sitting in the cell. It transports in 1 sodium. It exports in exchange with sodium out 2 protons. And also what's important, the crystal structures that are available are basically an apo structure because they were captured at pH 3.5 and at pH 4.5, which is, if you look at this, the inactive state of NhaA. Coming to the structure, you can see this is the unique NhaA fold, which is based on crystallography and cryo-EM data. You have a core domain and a dimer domain, which means if there's a dimer domain, which means NhaA forms an active dimer in the membrane. And studies have shown that cardiolipin is really important to form the dimer interaction, which is important for function. And you can see if you extract cardiolipin from the preparation, that the activity of NhaA is compromised. What you can see here, there are certain residues that are in this interrupted TM4. There's also interrupted TM11 here, and these residues are proposed to be interacting with sodium and protons. And if you go a little bit closer, there are certain residues that have been shown in functional studies that are either irreplaceable or are important for sodium and proton transport. So this is basically what has been known. And the question is, NhaA was analyzed by different groups. The Padan group in Israel was pretty leading the way into this research. Other groups, David, it's on the call today, also participated in this kind of research. And why me? Why would I do work with NhaA? So I've been as mentioned before, I worked with different importers, and those are in the NSS family, in the SSS family, and in other families, and all those that have a red arrow are the ones that I worked with. And you can see they transport what we consider the solute, which is important for the name, but they also co-transport either sodium and or protons and different ions here. So basically, I've been working with sodium-dependent transporters my entire life. Also, I was involved in analyzing the sodium binding site for PET-P, which was not clearly identified with crystallography. So I used a technique that I introduced in 2007, and this was basically using the SPA, which is the scintillation proximity assay. So Itana Padan asked me about 2 years ago if I would be interested in joining forces and analyze NhaA by analyzing direct sodium binding to NhaA. Why is this important? Because, as I mentioned before, there's an apo structure only available, and it's not clear specifically for NhaA, even though there are other sodium-dependent transporters, that the sodium binding site in NhaA is not clearly defined. So what I've been using is the SPA assay, and this works by using scintillation SPA beads. And the fact that NhaA has a his tag, we can use a copper chelate approach, basically by immobilizing the his tag protein to the copper coated beads. And you can see a scheme here. Those are the beads with the copper coating. And if you use a radial ligand, it'll be in solution and there's no energy absorbed by the bead. As I mentioned, if you have a his tag protein, the his tag binds to the copper coating. Once the protein binds its ligand, radio-labeled ligand, you get light emission. This is a very neat assay. It's very quick and doesn't produce much waste. So basically everything you can do is in a plate format. There is no liquid waste or any major waste accumulation that would prevent this. And this is specifically important for sodium-22, which is a very strong isotope, and you don't want to have any contamination in the lab. The original assay was developed for tritiated compounds, for iodide 125, C14, S35, different phosphates. I also extended this assay involving calcium 45, iron 55, and even, as I mentioned, sodium 22. The assay works in the same way. You have a copper-coated bead, you have your ligand. If it's not bound to a protein, like shown here, there's no light emission. You can interrupt this interaction of the his tag protein to the bead by adding imidazole. So it's a very nice format, and you can do on rates, off rates of binding, but you can also do equilibrium binding. And this is what I'm showing, what I've done for NhaA. What I've done here, I have first tested how much protein I need to do the assay. And I'm showing here the increase of purified NhaA. And you can see in red and white, this is wild type. And in yellow, this is one of the mutants that's located here. It's one of the CPA mutants, and this residue has been shown to interact with sodium. So this is kind of the proof of concept. If I would get activity with this mutant, I would expect something is wrong. However, everything works just as predicted, and with this mutant, there's no activity. As I mentioned before, cardiolipin was shown to be important for binding. So the data in red show wild type in the presence of 5% cardiolipin, and at a low detergent, in this case, it's 0.015% DDM. So you can see with increasing concentration of NhaA or amounts of NhaA, we basically get increased binding activity. What I show here is specific CPM, counts per minute, from the SPA beads. And basically it gives us a readout what amount of NhaA is useful for those assays. And basically, based on this curve, we choose about 200 nanograms of pure NhaA in those assays. So the next step was to test the effect of different cations on NhaA activity. So if we have no ions here, we get 100% activity. If we add 10 millimolar non-labeled sodium, we see a significant reduction. The same happens for lithium, which makes sense because lithium was shown to be a co-transported ion. Potassium does not inhibit the activity. Thallium, however, does inhibit. And if you look closer, we can actually plot the activity of sodium binding reduction by increasing concentrations of thallium chloride, and the affinity or the apparent affinity is in the 10 millimolar range. While thallium was shown to bind to a sodium protein antiporter. It's called NHAP from Pyrococcus, one of the Pyrococcus species. And thallium was used as the surrogate for sodium. So there is a structure available where actually thallium is in the binding site in this pocket here. So there's some identification of a sodium binding pocket using a replacement for sodium actually. So the other important measurement that I performed here is, again in yellow, this is the mutant D163N. There's no activity regardless of the pH. However, if we use wild type, and then in this case we have cardiolipin and low DDM available, which means you have at a high pH, pH 8.5 was described as the perfect activity range. So if you go to a pH of about 9, you get 100% activity. If you go down, the amazing thing that happened here, activity doesn't go down all the way. It stops at around 40%. And this indicates that sodium, even at low pH, is able to bind to NhaA. If we continued further and measured the affinity for sodium or the apparent affinity for lithium by replacing 50 micromolar sodium, 22 bound to NhaA, by increasing concentration of cold sodium or cold lithium, and under different conditions, this is the as purified condition where we have 0.1% DDM, we reduce DDM, here's high DDM with cardiolipin, and here is low DDM with cardiolipin, and you can see that the Bmax, not the Bmax, the binding actually increases with all those conditions, and that the affinity also increases, and this is shown in this plot. So the cyan blocks show basically the increase in the binding, and then in red you see the affinity for sodium, and at this condition where you have high concentration of DDM, the affinity for sodium goes from 2.3 to down to roughly 0.5 if you have low DDM and cardiolipin present. Lithium has a much higher affinity, and this helped actually before this high affinity apparently to run other binding experiments using ITC. So this is consistent with previous data, and those binding data also match the actual data that were collected in the Padan and other labs with regard to uptake activity and the affinities for sodium and lithium. So NhaA and solid supported membrane electrophysiology. In 2005, Klaus Fendler's group, in collaboration with Susanna Kadan, they also introduced this very nice feature, which is the basis for the SURFE that I'll be showing in a minute. As you can see, if you have NhaA in a liposome system, if you come from a sodium-free buffer, you change to a 10 millimolar sodium, you get a downward movement of your current, comes to a plateau, and then when you change the solution from sodium-containing to sodium-free condition, you have the reversal of the current. If you take the peak current here, the peak meaning from 0 to minus 1.8, we can plot the activity as a function of sodium. And this is done at pH 8.5 with two different liposome preparations. And in both cases, the apparent affinity for sodium is in the 10 millimolar range. This is about 20 times more than expected from cell experiments. And compared to the binding data, it's also reflects a lower activity affinity here. So the fact that I received NhaA, was able to reconstitute in liposomes, and I was trying to do some measurements with basically using sodium-22 flux. But then at this time, I learned about the SURFE, and since George with the NhaA team is just across the river here on the Jersey side, I drove over and tested out the SURFE. And I was amazed when I saw my first measurements. This is the very first measurements I've been doing. So if you have a control liposome preparation, there's no activity upon the change of sodium in the buffer. However, if you have NhaA, just like the original data that were produced in the Fendler lab, shows that you get a downward movement of the current, and then the reversal upon removal of the sodium in the solution. This is due to the fact that sodium enters the liposome and protons exit. And in this case, the net charge is one positive charge is exiting the preparation. As a result, you get a downward movement of the current. If I work with sodium-dependent systems, where it's only sodium going in, this curve basically goes up. But here, in this case, the net flux is one proton out, resulting in a downward movement of the curve. Since I'm affiliated with COMPA, the Center on Membrane Protein Production and Analysis, which is led by Wayne Hendrickson, we were able to purchase this machine and make this available to the scientific community. So if you're interested, please check out this website. And if you have any requests for testing the machine or having any proteins of interest that you want to have serviced, let us know. So since we have the SURFE now in our lab, we could measure sodium-mediated fluxes. And as you can see here, the same curve that I showed before, basically, when we have a preparation of liposomes containing sodium NhaA wild type, you see the red curve. And if we use the mutant, there's no activity, which is consistent with previous reports that this mutation impairs sodium-dependent proton flux. And as I showed before, there's also no sodium binding by this mutant. If we check the activity in this kind of measurement, so this was done at pH 8.5. And if you change the pH of our solution, you can see at pH 5, there's no activity, and it peaks around pH 8.5, which is consistent with previous measurements in the cellular system or in membrane vesicles, basically. When we take these measurements and we test sodium concentration effect or the effect of lithium on those currents, we can see that based on the charge transfer, we plot, as opposed to Klaus Fendler, he used the peak current, we used the area under the curve. So we plot the charge, basically current over time. And what you can see here, for sodium, we obtained an affinity in the 0.8 millimolar range. And for lithium, it's in the 0.1 millimolar range, which is consistent or very close to what has been reported with previous measurements using cells or membrane vesicles. So what are the next steps? As I mentioned, I don't think I have the mechanism that I could present to you. So the next step would be to mutate these different residues and analyze the effect of the mutations on binding and transport to get a coherent picture of how mutations affect the activity of the protein overall. Most of the work that was done in the lab was by Ava, which is a postdoc in the lab. Tisha assisted. Eva was a high school student who was around when the SURFE was delivered, so she worked a lot on the SURFE actually, and this also reflects how easy it can be to get this method running in the lab. Audrey, my technician, also helped. Justin just joined the group and is happily using the machine. Itana Padan and Manish basically helped just the preparation of NHAA, and we published our binding data recently. To the whole NhaA team, because if we have a problem, we just make a call and we get all the service we need. And then on the Compa side, Wayne Hendrickson and all the other members on the Compa team and the executive committee. Obviously, there's funding, which comes from NIH, different sources, and they're acknowledged here. So thank you for your attention.
Thanks so much, Matthias. David, now, if you want to share your screen, the floor is yours.
All right. Okay. Sorry, I'm talking next. I have to. Oh, great. Yeah. Great, Matthias. It was fun to see some of the older data as well. Let me just share my screen. Right. Right. You can see my first slide. Is that right?
Perfect. Yep.
Great. Well, thank you for everyone. I saw other people dropping in that I recognize. Hope you're all doing well this time. It's fun to see Itana there too. Hope you're good. So today I'm going to present some of our insights into quite an unusual sodium protein exchanger, I think. And I say mechanism, but like Matthias, I also agree that NHA and all these sums, we're still really, there's a lot of details that we still don't know yet. And in fact, this system has a lot we don't understand. But to put the picture, these belong to the small molecule transport, solid carrier transporters, which we called mailer systems, around 450, of course. And just to remember that a lot of them, because it's so important to cell homeostasis, mutations, a lot of them are linked to some form of inherited disease. And actually quite a number of these, of course, are linked to the transport of ions. So our lab is sort of the idea that if we can obtain structures, we can obtain a more mechanistic picture of how they're working really. And that we hope that information can help understand how these systems might be working in the physiological context. And so we work on both facilitative glucose transporters or GLU transporters and these sodium protein exchanges, which I'll talk about today. As Matthias mentioned, NHA is really has been the paradigm for these family of exchanges for many years, and that used the established protein motor force to export sodium. So conditioning the bacteria for sodium salt stress. Of course, there are bacteria that live in high salt conditions, and they also do electroneutral exchange. And this is generally what the mammalian systems do. They establish sodium gradients to exchange for protons. These are electroneutral. And these activities have really been very important to help maintain cellular pH and also in organelles, and also cell volume and sodium regulation as well. As Matthias briefly mentioned, there just to remind you, of course, that we have them everywhere, and we also have them in all compartments. And we have a number of different isoforms in humans belonging to the SLC9 family that you may have come across. NHE1 is a quite ubiquitous exchange of many cells, and others like NHE9, for example, is mainly restricted to endosomes and other organelle departments. the system I'm going to talk today is quite an unusual evolutionary member, SLC9B family, NhaA2, which is really uncovered by Regini and also Daniel Fuster at a similar time. And just to remind everyone again that this first structure that Vidal has solved together with Hartmann, Merkel, and Kroll-Hunter, they all have this core domain, which has these unusual helices with unwound regions which cross over near the central membrane. This is the only family that we see. This is now, it's called the NhaA fold. We've observed this structure in a bile acid transport. I believe Matthias also worked a little bit on that. So this fold is a sort of general fold that secondary active transporters use. Now this unusual feature of these unwound helices, and these create Lycal helical dipoles, which are neutralized by opposite charged residues. In this domain, we have the domain which is involved in oligomerization. And so we worked on a different homologue, a bacterial homologue in NhaA, and that ended up being a little bit more tractable than NhaA, and we could obtain a number of different conformations of the exchanger, which surprisingly showed these were now referred to elevator-like rearrangements of these transport domains. So the dimer domain acts as a scaffold. It does breathe in and out, you see, but the transport ions are carried across the membrane by only one of these domains. And so the real fundamental difference here is that the ion is only transported in this one domain, which can move against the other domain because it's fixed oligomerization. And more recently, we could obtain the first insights in the mail exchanger, and indeed they are quite an evolutionary conserved type of fold overall, particularly that the transport domain, as we'll present today though, and it's NhaA2, they have very differences in how oligomerization is taking place. So NhaA2 is, as I mentioned, Regini and Daniel Fuster characterized it, and they noticed, and indeed it's a little more evolution related to the bacterial electrogenic exchanges of, and Matthias mentioned NhaA. We've worked on NhaA from Thermus thermophilus than it is to its own sort of more NHEs. So it's quite a, if you like, unusual, and I think that the bioinformatics, if you like, based on the sequence context, it also fits its physiology. For instance, in flies, it's supposed to protect against sodium salt stress, indicating that it's exporting sodium, and indeed it does co-glycose the V-type ATPase. So it's supposed to, if you like, do the reaction that the physiological reaction that bacterial exchanges do, and that is used the proton gradient to export sodium. Another interesting feature is that they also have the two aspartic acids, which originally only found, or thought to only be found in electrogenic, at least only found in electrogenic bacterial exchanges. So the electroneutral exchanges only have one asparagine and one aspartic acid, so they don't have two aspartic acids. So this is interesting. So it has a similar relationship with the bacterial exchanges, and it has two aspartic acids. And as I'll mention here, though, there is a difference in the one of the residues that it salt bridges to. So I know for those people interested in CAR-EM and structural biology, I'm sorry it took a while. I think we spent several years trying to crystallize the system. And now I'm just going to present the results. I'm sorry I don't have time to go through that. It was challenging, like none of the systems are when they're so dynamic. But the interesting results here, of course, is as you can see here, you have this homodimer here, and the most interesting difference is you have this helix in blue, and this helix is unique to this family. In fact, NhaA2 has 14 helices. It has one more helix than what we see in the mammalian exchanges, and two more helices than what we see in NhaA. So this extra helix is actually domain swapped. So I've have the colors here. So this helix is actually part of this protomer. So it's not only domain swapped, but it's also separates the two domains by certainly a large degree on the cytoplasmic side. But if you just superimpose the monomer form against NhaA, which was bioinformatically one of the closer looking members, it actually superimposes really well. So the main difference here, of course, is extra helix TM1. And if you look at our NhaA, the homodimer, you can start to see the similarity there apart from this additional helix, which for some reason this exchange is acquired. So this helix, if you look at the contacts, it's actually not very strong contacts, just a small number of contacts between the other protomer. One is in a couple of the charged residues in this loop domain of TM7/8, and the other is some helix or hydrophobic packing, Van der Waals packing against TM8. So it's a small interface, which is also quite unusual as well. So of course we wanted to look at the why, of course, NhaA2 has this extra helix, and the first was just to see whether we could remove it, and we can remove it, and we can express it. And based on side exclusion profiles, it seems to migrate as a monomer compared to the dimeric form. And interestingly, just a mutation of this charged loop is enough to start to shift a fraction of this protein when you extract detergents into monomers, in fact. And in fact, this mutant and also TM1 is inactive in terms of looking at complementation under a yeast sensitive strain, which Olga sent us, and I see Olga there as well. So hey, welcome, Olga. Based on localization, we don't really see any differences in trafficking for these mutants. So we have these mutants which are required, at least we can say they're required for optimal activity, the dimer form. And of course, we wanted to look at this further, especially this unusual organization, and it's, should be lipid filled. So we sent some sample that Carol Robinson and Michael managed to fly. And we can see that the protein NhaA2 co-purifies with several lipids, at least the dimer does, whereas the monomers that fly through are essentially lipid free. And so at least we can say that, at least in ATMS spec, that the lipids prefer to stick, if you like, to the dimeric form. And we have a few masses here, and one of the masses match quite nicely to PI, and the other to PIP2, which is around 1042 Daltons. I don't have time to go into lipid screening, but we screen those lipids by thermal shift assays and things, and indeed we saw quite nice thermostabilization by those lipids. But I think this is probably the easy experiment just to show this. You can take NhaA2 and just, excuse me, lost my cursor here, just adding, even without heating, you see that the addition of PI to the purified form seems to help stabilize the dimer. And if we heat it up, the system, PID system, we see that the wild type protein, the protein without the extra lipids seems to dissociate to monomers, whereas a PIP2 indeed stabilizes the monomer, but also stabilizes the monomer, whereas PI, we see it specifically stabilize in the monomic form. At this stage, it's unclear whether it's because we're adding so much PIP2 in the system. I mean, it's sort of an artificial system, and we need to look with our lipids around, or in fact, so at this stage, we can say that at least confidently that both PI and PIP2 interact, whereas PI seems to specifically stabilize the homodimer. So we determined the structure of NhaA2 in nanodiscs made with pure PI lipids. So these are roughly, I can't quite remember, I think they're like 90% pure PI. Actually, we did mass spectrometry on those lipids as well. And they're consistent with the larger size masses that we're seeing from our protein, which is also purified in Saccharomyces, which I forgot to say, sorry. In any case, if we look at the structure, at least over the monomer and nanodiscs compared to the detergent, we see that it looks fairly similar. The major difference here is this extra helix that actually moved a bit. And in fact, that not completely surprising. I was just there are a number of proline and glycine residues before and after TM1, which makes this intrinsically effectable. If you look at dynamic elastic modeling, which we've done, we can see that indeed this helix is actually very dynamic. The interesting thing here is that the helix moves in, and we see very nice coordination of PI lipids at this interface. So we see really clear lipid remodeling, which we believe is induced by PI lipids. And as Matthias mentioned, in fact, this is really interesting because NHA also binds a negative, lipid interface. In this case, we could show that it binds carlipin. Again, the monomeric version is less. I don't show it here, but the monomeric is less sensitive to carlipin binding. Addition of carlipin to purify NHA shifts it from the monomeric to more the dimeric form upon heating. And indeed, we can mutate out these residues that are at this interface, this positive charge interface here, which is binding this negative charged lipid. NhaA nicely followed that up, but looking at cardiolation strain and the piece intention that indeed, in this deletion strain, addition of TDM shows that the dimer is indeed less stable when cardiolum is not there and it can't complement under the high stress conditions. So what does this mean? Well, I think it's quite interesting. I haven't mentioned, but NHA has a structural inverted repeat of 5 plus 2 plus 5. And generally, as sort of Lucy, Forrest was showing, that we can, these solid state contractiles always have these inverted structural repeats. So this is really the only family I'm aware of, at least, where we see an expansion in the structural inverted repeat. And so from NHA and its other exchanges, actually a 6 plus 1 plus 6. And in NHA2, we can define it as a 7 plus 7 with this extra helix now sort of somewhat related to the position of the linker helix. So this is interesting because the core domain is not changing, but the structure of dimer is changing. And I think putting this together really quite suggests that dimerization is a really important feature that has is evolved to change how they dimerize. And the differences we're seeing, lipid dependency suggests that indeed this is a way to start to regulate these exchanges through their sensitivity to dimerization. There was a little bit of a rush through the interesting structure, I think, and now I'm going to sort of look more like the mechanistic details in our use of SSM. And so, in fact, NHA has an unusual ion binding site. It's actually not very negatively charged. Haven't shown the figure here, but it's Ollie Bexton did some simulations. We see very few ions spontaneously get to the ion binding site. So it lacks some of the negative charges that we see in other exchanges. It's an outward-facing conformation like NHA, and it also lacks the neutralization of sort of at least negative charge ways dipoles. And on the other negative charge dipole, we have a lysine residue instead of arginine. So this is the ion binding site. We have these two aspartic acids I mentioned. On the other side, instead of a lysine in NhaA, we actually have arginine, which actually forms a unique salt bridge to a glutamate. Actually, something that Neurobiology had figured or thought from structural bioinformatics. So there's a unique salt bridge, and we also have this tryptophan residue here. And this tryptophan residue, at least it doesn't seem, at least in this conformation, we don't see any pi-cation interactions with arginine, but the arginine is interacting with the oxygen group in the backbone just before the tryptophan. In any case, we've got this sort of a little bit unusual binding site from what we've seen before. And of course, we wanted to look at the ion binding site and also the effect of mutations on that. So this results by Andre's group, I say Andre and anion, and then we ended up repeating it with our own SURFE instrument ourselves. So the first thing is we can show you with NhaA2, you get these very nice peak currents, which increase on addition of sodium ions. And as we'd expect, if you mutate out those ion binding aspartates, we see very little binding. Here we see the effect of pH on binding, so a slightly low pH on the extracellular side, we see this binding compared to at pH 7.5 and 8.5, and compared to, of course, the sort of dead mutant, we don't really see any pH dependent activity. Of course, then what you can do is exchange the sensors into different buffers and look at the effect of different driving ions. And what we could see here is that it can be driven by sodium or lithium, but we didn't really see an activity by rubidium or potassium. So it doesn't seem to be that potassium would be a substrate for this exchanger. Of course, we can look at the responses to lithium as well, and we see very large peak currents, which sort of surprised us actually, because this was fairly out of the, we sent the proteoliposomes to Andre, and this was, I don't know, but he said that it went very well straight away, and we have struggled actually with proteoliposomes for several years to get this to work, looking at pH sensitive dyes. So we're actually a bit surprised how well and how nice the signals are. And of course, we can see here, if you judge the lipid to protein ratio, then of course, as you would expect, you see a difference in the peak currents. And so we can look at the affinities just by changing substrate concentration and normalizing the peak current here in this case. And we see that we get a high affinity for lithium, around 5 millimolar at pH 7.5 and around 30-ish millimolar at 7.5, which is fairly similar to a lot of the exchanges in the cytoplasm. They would have a Km, I think it came around this range. Interestingly, the TM1 mutant, monomeric mutant, we can measure some binding, but it's obviously much weaker compared to the dimer form. So indeed, at least we assume this is binding and translocation of a charge, obviously, since we can measure it. So it seems indeed that the dimer is definitely helping to coordinate binding affinities, which could be related to, of course, the lipids. And I think it's interesting, Matthias results showing how carlipin is also helping to bind sodium. So stabilization of the dimer will help to bind the substrate. So this is a little bit more in detail in the ion binding site. I guess I don't have time for just move this down. I don't have time to go into much details, but so far we've just started to play with different mutations. This tryptophan is actually quite unusual here. It's normally a small non-aromatic side chain in this position, so this is quite unusual to have a tryptophan here. If you mutate that to phenylalanine, it's actually, we actually get a gain of function in terms of binding affinity, both for lithium and for sodium. In complementation assays, though, it's only functional for lithium, only functional for sodium, whereas lithium, we don't see complementation. So it's an interesting idea. Maybe it's binding with high affinity, but the turnover is compromised. And obviously this turnover is something we would like to be able to measure if you get the proteolome assays to work better. The other interesting mutant here is actually a salt bridge swap mutant. So individual mutants of those residues, of course, inactive, as you might expect, complementation assays. But this double swap mutation, in fact, is able to complement at least the lithium, but not for sodium. And actually, SSM, we can't detect any binding for this mutant for sodium, but in fact, the lithium affinities for this mutant are actually the same as wild type. So, lithium, as you might expect, is less strict in its coordination compared to sodium because more electronegative ion. But it's still a little bit interesting to me, in fact, that we haven't even perturbed the ion affinity for lithium. It's just solely sodium being affected here. We should say that that the complementation assays have been nicely consistent with what we see with SSM. Sorry. So lastly, and I think this is probably the more sort of interesting experiments, is to look at the activities. As you might have noticed, the currents are going in this direction, which means where a positive charge is translocated in, rather than charges going out in the other direction, as you expect for electrogenic transport like NhaA. And so if it's electroneutral, then lowering the pH on the outside should compete for the binding, and you should see less activity. And this is indeed the case. If pH is lowered on the outside here, we get much severe reduction signal. But if we lower the pH on the inside here compared to outside neutral pH, we only see a small reduction. So essentially, because sodium translocation we can pick up sodium translocation at a much faster rate compared to proton efflux, then essentially what's happening now, because this is under sort of symmetric pH condition, this is under symmetric pH conditions. So when we lower the pH on the inside, we create a proton gradient. So the sodium ions coming in exchange for protons more rapidly, and that's why we see a reduction in the total currents here. But so this is consistent with electroneutral transporter, even though they have two aspartic acids. I think that the key difference here is these aspartic acid is instead of a salt-bridged lysine in NhaA and NhaA, we have an arginine, which is actually quite separated in any case. But of course, arginine has quite a high pKa, which makes it a poor residue to bind and release protons at a neutral pH. And it would sort of consistent with an idea that the lysine is required for efficient proton translocation. Indeed, if you look at NhaA mutations and look at the effect drive the activities by membrane potential, it's really only histidine which retains this voltage sensitivity. So consistent with the requirement of coupling between a lysine and aspartic acid. So with that, just to summarize, what we've seen is quite an unusual way to oligomerize sodium protein exchanger. We see sensitivity to oligomerization to PI lipids. I didn't have time to go to the structure, but the maps for the PI are as good as the tryptophans, which is next to, we think that that's a regulatory switch. The first instance, NhaA2 is also found in intracellular compartments, which are enriched in PI lipids. And then this more compacted form, we think is a more active state consisting with transport activity that we see requiring oligomerization. And then we'll get a strict exchange for one proton per sodium, where the proton gradient is used to drive sodium efflux and constant with the B-type ATPase, where it's co-localized with. With that, I just mentioned that the postdocs who really driven the work, Roman and Ray, and my PhD student, Suk-Kyung Yong, who really been working the system. And a special thanks to Andre for finally allowing us to start to probe the function of NhaA2. And I did mention Olga for sending us this yeast strain too. And thank you for your attention.
Thank you so much, David. That was really great. And to Matthias as well for the conversation earlier. I think a lot of the people in attendance were quite active in the Q&A. So yeah, I'll just start from the top. Matthias, we received a couple questions during your presentation. The first, how is the orientation of NHAA in proteoliposome?
I can't say because we have not tested this. So I believe it's a random orientation.
And then a follow-up question, when you use the peak amplitude for analysis of SSM currents, do you get different KMs?
We have tested using the peak currents versus the charge, and basically both methods come to the same conclusion.
Perfect. And then while I let those in the chat add more questions for David, one more that came in through your talk. Matthias, thanks Matthias, super interesting. Why does NHAA have approximately 40% residual 22 Na+ binding left at low pH? Does it possess two Na sites? Are there two populations?
Well, I have a history with two sites. I'm very careful making a statement here. I think we can't rule out that the two sites and only one site is occupied. So if it's 40% or 50%, I think this is very close, which is within the error of within the range of error for those measurements. I can't really say if there are two sites. I wouldn't rule it out. It could be just like a partial occupation of the sodium site under those conditions. And also, this is important to note, this is at a 50 micromolar concentration of sodium. So this is not at saturating conditions. So as I said, I'm careful to say it's an option. We have like partial occupation, or there are two sides, but both are optional.
And then another question, perhaps addressing to both of you. Thanks to both Matthias and David. Great to see both of you. Do you think the lipid effect on substrate are about stabilizing a particular conformational state of the protein?
Do you want to go first, Matthias, or should?
Okay, so my question for Lucy is then basically, when she talks about the conformational state, is it like the activity of the protein overall, or if it's like a conformational state considered like an inward or outward facing condition?
Lucy, if you're still in the chat, I welcome you to join, or she's actually typing it now, so inward or outward.
I don't think it's like that the lipid actually prefers the inward or outward orientation. I think the lipid is just there to produce it's called a healthier protein that's more active. I think certain elements are not as smoothly running if certain elements like lipid are not present. This is my interpretation.
Yeah, I mean, I would certainly. I mean, it's something I actually haven't thought about. I mean, obviously, we've been thinking about oligomerization, and I mean, I think the NhaA2 is an interesting example because we have such a small interface, right? So the idea, elevators, of course, you have this scaffold domain, but they're generally fairly extensive. So NhaA2 is a system where we can actually manipulate quite easily and see just a couple of mutations, we can start to destabilize that. So, and it seems to be a system which is very sensitive to lipids, right, in terms of oligomerization. So I would, you know, and so I think it's, as I say, it's a regulatory step to help stabilize the scaffold, which would presumably facilitate a faster turnover of these transport domains.
Perfect. And then another question here, going back to something that was addressed earlier during your talk, Matthias. Once again, thanks, Matthias. I was interested in your SURFE signals, which were similar at 2 different LPRs. We saw something similar with the metal transporter in the past, but find it difficult to explain this. Did you find a reason why the current does not scale to the LPR?
Okay, let me just share my screen. I believe this is the answer to the question. Is it this picture, basically, this graph? Because this goes back to the original data from Klaus Fendler's lab in 2005. This is normalized peak current. So these may be actually different. So what we do when we measure different protein to lipid ratios and different amounts of liposomes, obviously the signal goes up. So we have different signals depending on the amount of protein on the sensor. In some cases, we can reduce substantially. In fact, NhaA is one of the fast proteins, so we can use very little protein. Other proteins require more amount in the measuring system.
And then another question for you, Matthias. How do you explain partial Na+ binding at high pH with a lack of transport? What does that say about the mechanism of transport?
I think this is inverse. It's not the high pH. It's a high proton concentration, so low pH. I think this is what I showed in the binding experiment. I believe that even at low pH, sodium can bind. So there's no replacement of sodium by protons. It's not a strictly regulated system where you have either proton or sodium binding. You can have binding of protons and sodium at the same time. And this would then be required if you change the pH, you get activity. How this sensing is actually working, I really don't know at this point. But then I believe once you reach this activation state, that sodium can pass as well as protons.
And then one for you, David. How do you decide the amino acids to replace the one in NhaA2?
How do I decide? Well, we just look at the ones that are obviously different and very conserved. Obviously, this is quite an interesting system because there's so many residues we could mutate. So the salt bridge is quite unique. So this is obviously, we want to look at that, and we was actually surprised that a salt bridge swap mutant worked. So at this stage, obviously it's affecting cation specificity, but we actually don't, we don't know why it's needed in this case. obviously, I mean, so we just mutated the residues that are obviously quite different, tryptophan, for example. So, I mean, I think I would say that the nice thing, I think the complementation of the salt-sensitive strain we see is seems to be very consistent, which is nice. It gives us an early handle on the system and the protein before we start purifying and start looking at it by SSM, because obviously you have to, purification, localized protein is certainly is more time consuming. But so far we've been very happy with the system, and it seems to be consistent.
And then we had a couple people join in late during the talk, David, so maybe this is something that you addressed, but which expression system did you use and which lipids for reconstituting the protein? Right,
so it's a good question. So it actually used Saccharomyces. And actually Saccharomyces, I think, ends up being, in fact, Regina used that originally to look at computation in NhaA-2. So, it turns out to be a very good choice, because Saccharomyces actually have quite a high PI content, about 30% PI lipids. And I should say we're struggling with the proteoliposomes assays for quite a number of years. We reconstitute ATPase, inconsistent activities. We actually used preferred brain lipids or soya lipids. We did lipidomics, actually the very small amount of PI lipids there. So we've since gone there back with the SURFE2R. I haven't shown you the data, but in fact the affinities are much higher in yeast lipids compared to brain lipids. So we're really seeing an effect on affinity with lipid composition, which would be consistent with our role of requiring these lipids for agglomeration. But this is also a plus for the system, right? Because obviously we want to be measuring turnover, and now we sort of get a better idea what lipids are required for those types of assays too.
Perfect. Yeah, so once again, I'd like to thank both of you for really great talks. I think everyone in the chat was pretty adamant and thankful, and they are all in favor for the great talks that you guys just had. And it looks like we're at the top of the hour. So I welcome anyone. If you didn't get a chance to ask a question, feel free to send that over to us. We'll pass that along to David and to Matthias, and we're happy to readdress that. Maria, if you'd like to say any closing words, I open the floor to you as well.
Yeah, thanks, Matthias and David, and thanks everybody for joining. I think that was really a great session. So have a nice day and nice evening.
Thank you.
Thanks so much, guys, for joining, and we'll provide the on-demand copy, and yeah, hopefully see you in 2022 in some iteration of this.
See you all. Thanks for going. Nice to see you. Those I mentioned. Take care now.
Okay. Bye now.
Bye.
Bye.